This is Atiyar Rahman from the University of Chittagong, Bangladesh. I'm welcoming you all to my presentation on plant products into therapeutics through a comprehensive and combined approach of experimentations. I'm indeed privileged to present in Emerging Scientist Conference ES 2022. Dear audiences, let me make a very short background on why we have conducted this sort of research. As you all might know that numerous plants have been used as natural remedies for therapeutics in many diseases including diabetes. Because of this, plants have low or no side effects. They are highly affordable and patient compliance. In our case, we try to do for diabetes, which is one of the alarming diseases, and currently 354 million of people over the globe have been suffering from diabetes and expected to be double by 2030. Currently, synthetic drugs on diabetes have numbers of side effects. They are expensive, and to some extent, they are incompliance to patients. Therefore, it's an emergence to work on plant metabolites and plant products to convert them into therapeutics. In our case, we have used Lasiya spinosa, a Bangladeshi medicinal plant, which is used by tribal communities for foods and some other uses. However, no evidences, no scientific evidences are available on the use of these plants. First and foremost, we tried to evaluate the antioxidative potential of Lasiya spinosa extract prepared by methanol and ethanol. As we know that oxidative stress is one of the pivotal factors to cause number of diseases, including diabetes. We did it through in vitro experimentations. And in vivo experiments, streptozotocin-induced fructose-fed diabetes model have been used in a 4 weeks intervention with 150 to 80 gram Western albino rats. After intervention, the animals are sacrificed and collected the bloods to separate serum. Organs are collected for histopathological analysis, as well as gene expression, and finally, we concluded with the network pharmacological analysis. In our experimentations, we found that Lasiya spinosa has very strong antioxidative potential in context of total flavonoid content, total polyphenolic content, lipid peroxidation inhibition capacity, inhibition of protein denaturation capacity. Also, we added DPPH free radical scavenging activity, ABTS assay, as well as iron chelating activity. All this together made evident to have very strong antioxidative potential of Lasiya spinosa. In animal experiments, animals' body weight, their food, drinks, their blood glucose levels were regularly checked. Oral glucose tolerance tests made the message that Lasiya spinosa increased the glucose load-bearing capacity of the animal with the streptozotocin induction. As we all know that the organs, particularly pancreas, kidney, and livers, are affected due to the diabetes, especially pancreas, which is found here that The effect has been mitigated, almost mitigated, by the use of Lacea spinosa. Organ-specific enzymes, alanine aminotransferase, aspartate aminotransferase, lactate dehydrogenase, creatine kinase, and the lipid profiles as well as creatine levels have been improved to attenuate the complications of diabetes induced by streptozotocin. Very interestingly, we found that the insulin secretion capacity made by Lacea spinosa is really promising. Lacea spinosa moderate dose has increased the insulin secretion, and the tissue architecture has been improved, particularly with the use of methanol extract. In case of the kidney architecture, we will score and we'll discuss the scoring pattern of kidney in our next slides. And we found as well the pancreatic changes. It is found that the methanol extract TM3 was significantly and very promisingly able to make a very positive impact over the islet of Langerhans cells of pancreas, which has been scored in context of islet cell necrosis, edema, inflammation, vacuole size changes, and regeneration. It's found that TM2 again was the most promising extract in these experiments. Finally, the antioxidative enzymes, superoxide dismutase, catalase, and glutathione peroxidase have been analyzed. It was found that superoxide dismutase and catalase were multifold increased with the treatment of Lacea spinosa, giving a message that concluded by the protein-protein interaction and network compound target interactions. At least 27 genes were regulated by the promising compounds of Lacea spinosa. And catalase and glutathione peroxidase genes are two of them, carrying the message that Lacea spinosa, especially the methanol extract of it, attenuated the diabetes and diabetic complications observed in the combined approach of our therapeutic development. The strong antioxidant potential of the extract might be the pivotal factor to achieve the goal through the regulation of the antioxidative genes, especially superoxide dismutase and catalase. However, long intervention time is very much expected to make the study more justifiable. So our next level approach is to isolate and identify the bioactive compounds, running the similar experimentation in a combined and comprehensive mode with the in vivo, in vitro, and molecular experiments for therapeutic development of using Lacea spinosa. I must thank and acknowledge the researchers of my love, as well as the Ministry of Science and Technology for funding the project, supports from the Department of Biochemistry and Molecular Biology, University of Chittagong, Research and Publication Cell for partially supporting the project, and finally, the Laboratory of Alternative Medicine and Natural Product Research of the Department of Biochemistry and Molecular Biology, University of Chittagong. Thank you so much, everybody, for your kind hearing and patience for joining us. Thank you very much.